Hello, and welcome to this series of instructional videos for MassAI. In this episode, we're going to look at how to work with MS1 data in MassAI. So far, we've been working only with MGF files, which contain MS level information and precious little on the MS level. We have a little bit of information with the precursor. But the precursor select for fragmentation happens at some point during elution, and the time picked may not be the optimal. It may not be where the concentration is at its best. We have no idea. So when it comes to quantification, we really need the MS1 level information. In order to work with MS1 level data in MassAI, we need to apply the filter that I described in a previous video. We will launch the filter. And in the interface, you'll notice this button here that says Convert MZXML to MGX. Now, MZXML is a very popular format for mass spectrometry data files. They contain information both on the MS1 level and also on the MS level. However, the MZXML is a compressed format and also encoded format, which means that it takes quite a while when you work with it to decompress it and unencode it every time you load the file. Also, it's very difficult to read into and correct any errors that might be in there because of the encoding. Therefore, we will convert it into a readable MGX format which contain exactly the same information as the MZXML, only in its decoded format. Let's do it straight away. We'll click this button and we will select our MZXML file. And we have run right here. We open it. And now the filter converts the file into a readable MGX format. Here we go. We can now close the filter. We have an extra file up here, so we have two files now. The original MZXML file, which weighs in at some 54 megabytes, and the MGX file, which contains exactly the same information, only it's somewhat smaller and it's actually readable in any text editor. Next up, we're going to conduct a search against this file. We'll launch MassAI. And just like any other search, we will select the file that we want to search against. In this case, it just happens to be an MGX file. Open it, as always. Define the search space. We have a few proteins that we know happen to be in here. So we load them in a FASTA file format. Proteases, this is standard triptych digest. Modifications, we'll stick with the defaults. We have reduced and calculated our sample. And that's all we have to do. Then we start the search. Now that the search has completed, we're greeted with the standard three windows in MassAI that present the results, starting with the identified proteins. Now in this particular sample, we're only looking for 4 individual proteins. The highest scoring protein is haptoglobulin, which means that it has a decent coverage. The fragmentation is good. We have plenty of observations on this particular protein, but good quality does not necessarily mean that it's there in the highest concentration. In contrast to a standard MGF search, we see over here an extracted ion count for the individual proteins. This is what happens at MS1 level. It's label-free quantification. Now these numbers tend to be relatively large and don't really necessarily mean anything. We are sometimes up to the 1 billion counts, which is difficult to read. So in the column next to it, we have the relative extracted ion count. Sorted by it, I will find that the C163A, human scavenger receptor, may not be the highest scoring protein, but it's definitely the one that happens to be in the sample at the highest concentration. Switch to the 4th tab sheet. I can see how the quantification of the 4 proteins match up. We work with a normalized concentration. So here we see the most present protein in the C163 is at 100%, where the haptoglobulin, which was the highest scoring protein, is down about 45%, half of the concentration. This is what we can do at MS1 level. We'll shut this down, and then we will look at the result list scan by scan. Select any peptide, and we get the annotated scan along with the annotated peptide sequence. But the interesting part here is actually this window over here, the new window, which shows us the information that we can gain at MS1 level. The scan that we see over here is the MS1 scan, which contains the precursor that has been selected for fragmentation. It's right here. It's highlighted with an arrow pointing downwards. We can go through the MS1 scans that contain the parent precursor and see how it elutes over time. The green one up here is the one that was picked for fragmentation. You see the green down here as well, and we will notice that this is the elution curve. This precursor is present in 5 MS1 scans, and the first one here is where it was picked for fragmentation. But as you can see by the count, it's probably not the most optimal place to pick the precursor, because the instrument should have waited just a few milliseconds extra to pick at this place. It didn't happen so. When you work with an MZXML file or MGX, MassAI will automatically populate this column over here for the extracted ion count. And this is a quick and easy way to do a label-free quantification of your peptides and also of your proteins. And that brings this episode to conclusion. If you want to stay updated with the latest videos, be sure to click the subscribe button. If you like what you see, click the like button. And if you have any requests for videos that I should do in relation to MassAI, be sure to leave a comment below. I'm your host Morten Rasmussen, and I'm the developer of MassAI. Until next time, happy searching.